So, hello students, welcome back to your own channel Vedantu NEET English. This is Basavaraj Sir, your biology master teacher. How is everyone doing, students? How is everyone doing? Doing great, amazing, okay, bad. How is everyone doing? Because today's class is going to be very special. Today's class is going to be very special because it is genetics class, that is numericals. Now, I know all of you might be wondering, sir, theory is done. Which type of numericals can come in examination? So students, in this session, you will be solving so many numericals based on directly on monohybrid, dihybrid, incomplete dominance, disorders as well as pedigree analysis. All types of numericals from today's class, even linkage, yes, even linkage from Southern. So all those numericals in one session. Apart from that, let's start the questions with NEET 2024. So all of you have finished with the principles of inheritance chapter, right? If you have not watched, please try watching the part 1, part 2 of the principles of inheritance. It is an RNA batch. RNA batch, part 1, part 2 principles of inheritance. Now, whatever you have learned there, now you will try to use it here now. Shall we start, students? Let's not waste any time. Let's just start at this point, right? Now, students, let me know in the chat if I'm audible and visible also, right? That is very important, obviously. If I'm not audible and visible, I'm talking to myself from past 1 minute then. Yes, I'm audible and visible. Ravindrani, Madhumita, yes. Vaishnavi, Divya, I think I'm visible to all of you, right? Yes, I am visible, right? And I can see green hearts in the chat. Let's keep up the green hearts in the chat because that's how you know, right? You are excited for botany classes. That's how I get to know that you are excited for botany classes with the green hearts in the chat, right? Green hearts in the chat. Okay. So let's go to the first question. Let's not waste any time. Let's go with the first question. Okay. Now order is going to be very simple. We need to get our brain charged first. Right. Before you solve numericals, you need to get your brain charged, active for solving numericals. How do you charge your brain then? The simple way to charge your brain is by solving some questions which are very new. For example, let's start the class with NEET 2024 questions, shall we? The recent PYQs, this year PYQs, let's start with those questions first. Shall we go? Let's go. The first question on your screen. Now students, every single question I will give you 20 seconds. 20 seconds for every single question. So your time starts now. Read the question and start. Okay? Read the question. 20 seconds for every single question. Okay? You're getting 20 seconds. Okay? Now match the it's a NEET 2024 question, students. It's a NEET 2024 question. 2024 question. Go. Okay. Two or more alternative forms of a gene is called as your allele, right? Allele. Okay. So A is going to be 3, right? A is going to be 3. Alternative form of a gene is called as an allele. So where is A is 3? Oh, only one option. What is this comedy? Huh? Only one option is correct. So option number 2. Option number 2. Only one option is correct. Option number 2. So if A is going to be 3, B is going to be 4, right? B is going to be 4, A is going to be 3. Cross of F1 with any of the following, that C is going to be 1, as well as D is going to be 2. D is going to be 2. Easy question. Students, I want everyone to keep a track of your marks, right? In the chat, every single time you answer a correct question, I want you to type +4. If you do one more question right, +4. That is 8 marks, 16 marks. So I want you to keep a track of all the marks in the chat today. Will you do that? All of you will do that? Yes. All of you do that. So all of you +4 in the chat. All of you were able to answer this question. +4 for all of you. +4 for all of you. Next question. Okay. I'll give you 20 seconds. Your time starts now. Read the question and then we start. Okay. Which one of the following can be explained as the basis of Mendelian law of dominance? Mendelian's law of dominance. Okay. Out of one pair of factors, one is dominant and other is recessive. Okay. Alleles do not show any expression and both the characters appear as such in F2. No, both do not appear, right? Both do not appear. Both appear, but they do show expression. Okay. Factors occur in pairs. Yes. Discrete unit controlling a particular character is called as a factor. Okay. Expression of only one of the parent character is in the monohybrid. All of you. Answer in the chat. Answer in the chat quickly. I will give you time. 20 seconds. 20 seconds time. Now some of you were like, board is blur. Students, tell me in the chat, is the board blur or Samyukya, increase the resolution? Is the board blur? No, I don't think so. What is the answer? To all the students who are confused with this answer, let me show you here. Obviously, answer is 1. The answer is 1, but listen here. Characters are controlled by a discrete unit called as factors. Point number 1. So, can I tell, where is it? Point number 1 is going to be correct here. Correct? Yes. Factors occurs in pairs. Discrete called as factors here. Here. This is the point. Then we have factors occur in pair. A dissimilar pair of factors. One is dominant, other one is recessive. One is dominant, other is recessive. Yes. The last point here. Can you see? The law of dominance is used to explain the expression of only one of the parental characters in the monohybrid cross in F1 and the expression of both in the F2. So, in the monohybrid. So, what is the answer? A, C, D and E. A, C, D and E. All of you who are able to say the answer, +8 in the chat, all of you. Keep your track of your marks. I cannot keep a track of all your marks. Simple as it. Okay, +8 in the chat. Next question. A pink flowered snapdragon plant was crossed with a red flower snapdragon. What type of phenotype is or are expressed in the progeny? Okay, time starts now, go. Time starts now, go. While you solve the question, I will also solve simultaneously here. Okay, simultaneously I will also solve. So, a pink color flower, right? Pink color flower is what? Capital R, small r. Okay, and was crossed with a red flower. Red flower is capital R, capital R. Okay, red flower is capital R, capital R. Now we make the gametes. Here. We have capital R, small r. Here we have capital R. Okay. Cross it. What do we get? Capital R, small r, capital R. Capital R, capital R, capital R, small r. So what is the answer, students? We get this is basically red and this is basically pink. So what is the answer? Red flower as well as pink flower. Yes, red flower as well as the pink flower. Option number 1. Now students, this is the third question, I believe. This is the third question from NEET 2024. Third question. Do you know how many questions came from this chapter? Do you know how many questions came from this particular chapter in your NEET 2024? Approximately 14 questions. 14 questions came from this chapter. Now you tell me how many marks. Tell me how many marks now. 14 questions means. Right? Right answer. Next question. Okay. In a plant, students, all of you are able to see, right? All of you are able to see? Yes or no in the chat quickly? In a plant, black seed color, that is capital B or capital B small b, that is one is homozygous dominant, other one is heterozygous dominant. Homozygous dominant, heterozygous dominant. This is basically the genotype. This is the genotype. Okay, genotype is dominant over the white color seed. Then we have, this is dominant. We have small b which is recessive, which is going to be recessive in nature. Okay. In order to find out the genotype of the black seed, you're trying to find the genotype of this. You're trying to find the genotype of capital B or capital B small b. Which one is it? Black seed. Which of the following genotype will be used? So basically, students, this is a typical example of test cross. In test cross, what do? In the test cross, we take the double recessive parents and cross it with the dominant phenotype. Right? So what is the answer? Double recessive parents is going to be capital B, small b here. Similar example is given here also, students. This is the two genotypes, capital W, small w. Similar to here. Similar to what we learned here. Now what do we do in the test cross? In the test cross, we take the recessive parents. Right? So answer is going to be what? Small b. Next question. On your screen, your time starts now. Go. The time starts now. All of you quickly know your marks also. Yes, I am from Karnataka. Kannada baratte. Sarina. Okay, next question on your screen. Next question. Match the following, list 1 and list 2. Okay. So all of you know Down syndrome. Down syndrome is the trisomy 21, right? 21st chromosome. So A is going to be 3. So 2 options are gone. Okay. Then alpha thalassemia. Alpha thalassemia. Very easy. Beta thalassemia also easy. But if you do not know between alpha and beta, you can always go for Klinefelter syndrome. Klinefelter syndrome is a type of your X chromosome, right? X chromosome. Klinefelter syndrome, extra X chromosome is there. So D is going to be 2. If D is going to be 2 and A is going to be 3, there is only one option left. There's only one option left. Yes? There's only one option left. That is option number 2. Students, do you realize in examination under extreme pressure, there is a very high chance that you might forget about alpha and beta thalassemia. Okay, it can get a little confusing, but you are very sure about your Klinefelter syndrome, which is going to be happening because of XXY condition. You know Down syndrome because of your trisomy of 21st chromosome. So if A is going to be 3 and D is going to be 2, only one option is correct. This is called as elimination technique. This is called as elimination technique. So tell me your marks in the chat quickly. What are your marks? And students, quickly smash that like button, all of you. Quickly mark your attendance if you're new to the channel and smash the like button, because obviously this is the Guru Dakshana which we take, right? Smash that like button. Now, next question. Go. This again, students, all of these questions are NEET 2024 questions, nothing new. Okay. I'll be solving the questions. I want you to solve the question with me. As per the ABO blood grouping system, the blood group of father is B positive. Okay. Let's write down. Father is going to be B positive. Father is B positive. Okay. And mother is A positive. Mother is going to be A positive. Okay. Mother is A positive. Okay. And the child is O positive. The child is going to be O positive. Now they're asking you the respective genotype can be. What is the respective genotype? Students, tell me, if the child is O positive, is there a chance that father or mother both can be homozygous dominant? Is there a chance that this father can be IB and IB? No, because this O cannot be produced then. Is there a chance that this mother can be IA and IA? No, not possible. Not possible at all. Right? Not possible at all, because if both were IA, IB, IB, this child will be AB positive. This child will be AB then. So since the child is O, father has to be IB and I. Mother has to be IA and I, small I. This small I and this small I will make up our small child here. Will make up the small child. Right? Small child. So what is the answer now? What is the answer? This is going to be there. Yes. Any other possibility? IB, IB, no, not possible. IA, IB, IA, IA, not possible, because child is going to be O. Not possible. Not possible. So only A is the answer. Only A is the answer. Did you realize? Only A is the answer. Very simple question from NEET 2024. All of you are able to solve. Yes. Be confident in your answers now. So only A. It has to come here. This has to come here. Only A is the answer. Only A is the answer. Okay? Only A is the answer. Nice. Next question. Next question. Again, all of you, quickly tell me your marks in the chat. Keep your track of your marks. It is very important. Keep a track of your marks. Because I want to know at the end of it, I want you to comment in today's video how many marks you got. That is the today's trick. At the end of today's video, I want you to comment down what is your marks out of this genetics. Because if this session is going to be really good and helpful to all of you, we can make a similar session of all the numericals in biology. What do you think? Numericals from your cell chapter, numericals from biomolecules, numericals from the reproduction, that is sexual reproduction, flowering plants, gamete questions. All the questions will keep a track. Okay. Next question. ABO blood grouping is an example of your multiple allelism. Multiple allelism. Easy question. Easy question. Marks in the chat. Next question. A cross between a tall plant was made, resulting in an offspring of tall and dwarf plant with a 3:1 ratio. The genotype of both the parents is. Very simple question, students. Very simple question. Very simple question. Very simple question. Yes. A cross between tall plant was made, resulting in a tall and dwarf plant with a 3:1 ratio. Students, Just tell me what do you cross in F2 usually? That's all is the answer. What do you cross in F2 usually? What do you cross in F2? That is going to be your answer. That's all. It's very easy. In F2, what do you do? Capital T, small t, capital T, small t. That's how you get 3:1 ratio, right? That's how you get 3:1 ratio. Simple. Simple. Such an easy question. Such an easy question. In your dihybrid, in a monohybrid cross, you do F2, what do you cross? Capital T, small t, capital T, small t. Selfing of your fun. Selfing of your fun, right? Selfing of your fun. Next question. All your marks in the chat also. If you're going wrong, tell me you're going wrong somewhere. Don't lie and all. Okay? Don't lie. Lying is bad. Okay. Next question. Which of the following is not an autosomal recessive disorder? Not a autosomal recessive. Now students, how do you remember this? Like, how can you remember all this? Is it easy to remember this? Yes, it is very easy to remember. How do you remember? Let me teach you. Here. This paragraph. This one. And you can see I am also here. I'm second. I am also here. Two places. Okay. So genetic disorder, remember I made you write this down. Under Mendelian, autosomal dominant, only one example, myotonic dystrophy. Under autosomal recessive, we had phenylketonuria, sickle cell anemia, and thalassemia. Sex-linked, we had color blindness and hemophilia, right? And in chromosomal, we had Down syndrome, Turner syndrome, and Klinefelter syndrome. So what is the question saying? The question is saying not a autosomal recessive. Autosomal recessive, only three are there: PKU, CSA, and thalassemia. So what is the answer? Which one is missing here? Thalassemia, PKU, sickle cell anemia. Only which is missing is hemophilia. So students, this is how you solve questions. You make a flow chart, remember the flow chart and solve the questions. And students, if you want this PPT, right, after end of today's class, you want this presentation, where do you get it? All of you know it already. That is on Telegram channel. All this entire presentation will be available on the Telegram channel. Okay, also on the description of the video. Here on my Telegram channel, this entire PDF will be available today only. Basavaraj Sir Biology. Telegram channel will be available to all of you. Okay, don't forget. Hemophilia is the answer. Hemophilia is the answer. Okay. Next question. Next question. Students, all of these questions are NEET 2024 only. Marks in the chat, all of you. In the given below are two statements. Okay, assertion. In honeybees population, son do not have father but can have grandfather. True. They do not have father. They do not have father, but they can have grandfather. Yes. They do not have son. They can have grandson. I've taught you this. I've taught you this. Right? How many questions? More than 50-60 questions are there today. Don't worry. You will be satisfied by the end of today's video. More than 50 questions are there. You chill today. Just keep solving. Now, haploidy in drone of honeybees is due to parthenogenesis, while fertilization results in the female bee. Yes, whenever there is fertilization, it will result in a diploid structure, which will invariably be a female. It will be a female. And how are the males produced? Males are produced by parthenogenesis, where males are usually haploid. Okay, haploid. So what is the answer? A and R are true, and R is the correct explanation of A. Easy question. Students, do you remember this? I've taught you this in my class. In my class, I've made you learn this. Can you see? This is called as honeybee 1. See, mitosis, we get haploid. Meiosis, we get gamete. Why? Why mitosis? Because the parent itself is haploid. So mitosis, the gamete. Parent is diploid. So meiosis, we get the gamete. Fertilization will result in always in a female, and the female can be worker or queen. How is the male produced? Male is produced by parthenogenesis directly from the egg. Directly from the egg, male is produced, which is going to be called as drone, and it is haploid in nature. And did I make you write this also? Did I make you write? Cannot have a father, cannot have a son. It can have a grandson, it can have a grandfather. My notes. Learn this. Okay? Next question. Again, question from NEET 2024. 14 questions, students. 14 questions came in NEET 2024. And all the questions were very easy. And easily you would have scored above 50 plus if you have attended my class. Easily 50 plus. Read the question. A yellow seed and a violet flower. A yellow seed and a violet flowered pea plant was crossed with green seeded and white flower pea plant. The progeny showed plant with 4 different phenotypes. The yellow seed and violet flower must be. What should they be? Read the question, understand the question, then solve. Understand the question and then solve. What should they be? Yellow seed and violet flower pea plant. Yellow seed and violet flower. Yellow seed and violet flower. Right. Yellow seed as well as violet flower. Yellow seed and violet flower. Right. Let's write yellow seed, violet flower. Okay. Was crossed with green seed and white flower. Green seed and violet and white flower. Okay. Now the progeny showed plants with 4 different phenotypes. The progeny were 4 different phenotypes. 4 different phenotypes. Okay, 4 different phenotypes. The yellow seed and violet flower parent must be. They're asking about the yellow seed and violet flower. Students, they're asking you how the parent looks like. How does the parent look like? Now tell me, simple. Let's do simple. What if the parent is heterozygous condition? What is it going to be? Capital Y, small y, capital V, small v. Yes, capital V, small v. Do the cross now. Do the cross. If it is going to be heterozygous, what are the gametes we get? Tell me. The gametes which we get is what? Four different gametes. Yes, 4 different gametes. Now you cross it with the other parent. Other parent is homozygous, normal. Okay. We end up getting 4 different phenotypes. So the students, when you get such questions, right? It's a trial and error. It is a trial and error. You're supposed to try and then get the answer. Try and get the answer. Okay. Try and get the answer. Double heterozygous. This is called as double heterozygous condition. Double heterozygous. Imagine it was completely homozygous. If it was completely homozygous, what would happen? If it was completely homozygous like this, we end up getting only 2 gametes, right? We end up getting only this. Now you cross it with the other parent, you will not get 4. You will not get 4 different phenotypes. 4 different phenotypes is only possible when it is a heterozygous condition. It is a trial and error condition. You are supposed to try this question and then get the answer. Do all of you understand? All of you understood? Understood? Next question. A phenotype, again 2024 question. A phenomena where a single gene exhibits multiple phenotypic expression is called as? Quickly in the chat. And if you get -5, write -5 also. 1 marks you lost because attempt and 5 marks. All of you. If you don't attempt also, tell me if you don't attempt also, okay. A phenomena where a single gene exhibits multiple phenotypic called as pleiotropy. This is easy question. Pleiotropy. Direct question. This is direct question. Okay. Now students, again NEET 2024 question. Okay. In animal kingdom, along with XXYY mechanism, many other variants of chromosomal sex determination mechanisms have been described, such as XXXO mechanism, ZZZW mechanism, ZZZO mechanism, and haplodiploidy mechanism. Which of the following present respectively? Respectively, they are supposed to ask. Okay. First, you tell me XXX0. XXX0 is a male heterogamety, right? Which is also seen in the case of humans. I'm not talking about the chromosome type, not XXXY. I'm talking about male heterogamety. Is seen in the case of grasshopper, right? Then ZZZW mechanism, ZZZO mechanism. Quickly in the chat, I'll give you 20 seconds to answer this question. Take 20 seconds and answer. I don't want to spoil the answer for you now only. Students, do you remember this? Do you remember this? When I taught you recently, I told you under male heterogamety, we have humans XXXY. Grasshopper is XX and X0. Both male determines the sex of the chromosome. Male gametes. While in the case of your female heterogamety, we learnt about birds are ZW and ZZ. Butterfly are Z0 and ZZ. Butterfly or moth. Butterfly or moth. Okay, butterfly or moth. Now tell me the answer. If bird is going to be ZW, grasshopper is ZXO. Grasshopper is XO. Tell me the answer now. Grasshopper, bird, moth and wasp, right? What is the answer now? Option number B. Exactly. Understood? Students, it is very important to write down in a fashion which will help you to remember also. All of these notes are already available in the Telegram. You should know that. Okay. Next question. Again, NEET 2024 questions. Many questions are there. The manifestation of phenylketonuria are given below. Okay, these are the symptoms of phenylketonuria. Choose the incorrect answer from the given options. Phenylketonuria, all of you know, it is a type of polygenetic inherit. It is a type of your polygenetic pleiotropy. It is a type of pleiotropy, right? One gene, multiple phenotypic expressions. One gene, multiple phenotypic expressions. Pleiotropy. Now tell me which of the following is not a symptom. Mental retardedness is, skin rashes is, accumulation of phenylpyruvate in the blood is, but furrowed tongue, no. Furrowed tongue is not a symptom. Furrowed tongue is an example of Down syndrome. Down syndrome. So option number 4. Option number 4. And that's it, student. That's it. That is the end of all NEET 2024 questions. How was it? Tell me your marks right now at the end of NEET 2024. At the end of NEET 2024, what is your marks right now? Quickly in the chat. What is your marks in the chat? At the end of NEET 2024 questions. Yes, good. All of you are somewhere in the same range except one question which was a little tricky because that requires more time. I think I did not give you more time. But apart from that one question, all of you are able to answer very well. Right? Nice, nice. Now, let's go with the next now. Next. Can we start now, students? Now from here we will do some numericals. As promised, we'll do some numericals. The first numerical is on the board right now. First numerical is on the board right now. Okay. Simple question. In a cross between male and female, both heterozygous for sickle cell gene, what percent of progeny will be deceased? Both are heterozygous. In a cross between male and female, both are heterozygous for sickle cell anemia. Both are heterozygous for sickle cell anemia. Male and female. What is the percentage of progeny? So write down male here. So while I'm solving, you try to solve on your own. We have female here. Male and female. Both are heterozygous. Both are going to be heterozygous. That is HBA and HBS. Female is also going to be HBA as well as HBS. Now let's do a cross to find the progeny. Let's do a cross. We have HBA, HBS, right? Instead of doing it, let's do it from here only, it's easier. I'm not getting space to write here. One second. I'll do it as such only. This and this, this and this, okay. This and this. That's all. What is the answer? Tell me. When you cross this with this, and this with this, and this with this, and this with this, this will be completely normal. This will be carrier. Second progeny also carrier. Only one will be affected. When both S are present, it is going to be affected individual. So out of 100%, if 1 out of 4 is basically 25%. It's going to be 25%. How many of you are able to write this right answer? All of you understood how it is done? If not, quickly ask me. If not, quickly ask me. All of you understood? Male and female. HBA, HBS, HBA, HBS. Both are heterozygous in nature. Both are heterozygous in nature. So what we do? We did a simple cross to get the. You can also do the simple that cross also. You get the same answer. You get the same answer only. Okay. Now since I didn't have space, I did it here. So what did we get? HBA, HBA, this to this, HBA, HBS, HBS, HBA, and HBS and HBS, hemoglobin S. So when that happens, that is basically 1 by 4, that is 25% is the answer. 25% is the answer. Next question, go. Read the question, 20 seconds, your time starts now. Marks also in the chat, marks also in the chat. I want to know the marks. In Snapdragon, a red flower was crossed with a white flower. Red flower was crossed with a white flower, and in F1 generation, the pink flower was obtained. When the pink flower was selfed with the F2 generation, showed white, pink and red. Choose the incorrect statement from the following. Which is the incorrect statement from the following? This question is with respect to the incomplete dominance. With respect to the incomplete dominance, all of you. Law of segregation does not apply in this experiment. This experiment does not follow the principles of dominance. This experiment in F1 is due to incomplete dominance. Yes, the ratio of F2 is 1 is to 1, 1 is to 2 is to 1, red, pink and white. Incorrect statement, incorrect statement. Absolutely, the first one. Why is it incorrect, students? Because remember, your law of segregation is universal. Law of segregation is universal. There are not exceptions here. There are no exceptions here. Understand that. Law of segregation is universal. There are no exceptions to this. Right? So, everything is correct. Right? Everything 3 are correct apart from the first one. Students, are you understanding? These are difficult questions now. These are some difficult questions which I have gotten for you. Solve these questions, you will feel more confident. And you will know where you are going wrong. Okay? So, law of segregation is universal. Next question. What is the expected percentage of F2 progeny? What is the, very good question, understand this question. What is the expected percentage of F2 progeny, okay, with yellow and inflated pod in the dihybrid experiment involving pea plant with green color inflated pod and yellow colored constricted pod? Okay, very simple question. I will explain in the most easiest way. So understand this, okay. Tell me first of all, what is that in your simple dihybrid cross, which of the following traits do you take? Tell me, which of the following traits you take? You usually take seed shape and seed color. We usually take seed shape and seed color? Yes or no, quickly? In a typical dihybrid cross, we take seed shape and seed color. We take seed shape and seed color. Yes, we take seed shape and seed color. Typical dihybrid cross. Seed shape and seed color. Okay, seed shape and seed color. In a typical dihybrid cross, what do we take? We take round yellow. We take round yellow and wrinkled green. Round yellow and wrinkled green. Typically wrinkled green. Now what is the ratio we get? We get 9:3:3:1. Right? Out of 9:3:3:1, students. In this 9:3:3:1, what do we take? We take this 9 is going to be round and yellow. Round and yellow. This 3 is going to be round and wrinkled. Round and wrinkled. Yes. Round and wrinkled. What is this? One more 3. This 3 is basically wrinkled yellow. Wrinkled yellow. And this one more 1 is going to be your wrinkled green. It's going to be wrinkled green. Now this much all of you know. This much all of you know in the typical dihybrid cross. Right? Typical dihybrid cross. Now in this question, what are they asked? In your question, they have changed the trait. They have changed the character. Instead of seed shape and seed color, we are taking pod shape and pod color. Can you see? We are taking green colored inflated pod. We are taking green color inflated pod. We are taking green color inflated pod. Inflated pod. And we are taking yellow colored constricted pod. Yellow color constricted. Yellow constricted. Yellow color constricted. Now again for this experiment also, if we do the dihybrid cross, we end up getting 9:3:3:1 ratio? Yes or no? Students, why am I going so slow on this? Because you need to understand they can change the character, they can change the trait, and they give you a question. They can give you a change in character and they can change your trait and they can give you the answer. Okay? They can give you. Okay? They can always change and give you. They can always change and give you. Now here what do we have? We have green inflated. Similar to this, we have green inflated here. Green inflated, most dominant. This thing is going to be what? This will be same. Green constricted. Green constricted. Now what do we have here? Instead of wrinkle, we will have yellow color. Yellow is wrinkled here. Yellow. Yellow inflated. And finally we will have yellow constricted. Similarly, this is how you should know. You should be able to tell each and every type of 9:3:1 ratio. Now what are they asking you? Question. They're asking you progeny, F2 progeny with yellow and inflated pod. Yellow and inflated pod. What do we have here? Yellow and inflated pod. Do we have here? Yes, we have here. Yellow and inflated pod. Yellow inflated. Yellow inflated. We have here. Okay. We have the yellow inflated here. Yellow inflated. Now, what is the formula? How do you find this, students? How do you find this? Progenies. Very simple formula to find the progenies. That is number of progenies divided by total progenies into 100. That's all. Do 3 by 16 into 100. 3 by 16 into 100 will approximately give you 18.75%. 75%. Okay. So do we have that option? Yes. Option number 3. Option number 3 is the right answer. Students, like this, they can ask you many questions, so you need to know which one exactly which you need to approach. Understood? So you need to always look for specifically. Now imagine, for example, I will ask you next time in the examination, instead of asking yellow inflated, they might ask you green inflated. That time what should you do? If they ask you green inflated, all you need to do is 9 by 16 into 100. That's all. Which is roughly around 56%. Roughly around 66%. Understood? That's what you should do. This is how you should do. Okay? Beautiful. Next question. Okay. A low frequency recombination indicates that the genes are low frequency. A low frequency recombination. Recombination is very low, means linkage is very loose, right? Located far on the located far from each other, located close to each other. Genes are located very close to each other. Not linked, present on a different chromosome. If recombinant frequency is very low, it means the linkage is very high, right? If the linkage is very high, means what? Understand. Students, all of you know, right? If linkage is going to be very high, recombination will be very low. Recombination will be very low. Right? Recombination very low. Now here we have recombination is very low, linkage is very high. Now what is the meaning of linkage? Means why? The genes are tightly linked. Yes, the genes are tightly linked, and meaning of tightly linked is that they are very close to each other. Understanding? This is how you answer the questions. Okay. So genes are very tightly linked on a chromosome. It means they are very close to each other. Genes are very close to each other. Next question. If a colorblind female marries a man whose mother was also colorblind, what are the chances of her progeny having colorblindness? Very beautiful question. A colorblind female, okay, right? Colorblind female marries a man whose mother was also colorblind. This guy is there whose mother was colorblind, right? Mother was colorblind. The X chromosome is coming here. If the mother was colorblind, this is the mother. This is the colorblind female. Marries a man whose mother was also colorblind. So mother was colorblind, the same X chromosome is coming. Now, what are the chances her progeny is having the color blindness? Now do the cross. What is the percentage now? Tell me. What is the percentage now? All of them? 100%? Understood? 100%. Beautiful question, which can trick you sometimes. Tell me your marks in the chat, all of you, quickly. Tell me your marks in the chat. Understood? Understood, students? Should I repeat again? No, right? See here. Female is colorblind. This is the female. This is the male. Male's mother is colorblind. Same X chromosome has to come. So he also will be colorblind. So both male and female both are colorblind. The offsprings, all of them will be colorblind only. Beautiful question. Numericals are very nice in this chapter. Next question. A heterozygous pea plant with violet flowers. A heterozygous pea plant with violet flowers. Okay, heterozygous. It is heterozygous. Heterozygous plant with a heterozygous pea plant with the violet flowers is crossed with homozygous pea plant with white flowers. Homozygous with white flowers. Okay. Violet is dominant over white, obviously. Which of the following represents the expected combination of among the 40 progenies or 40 offsprings? What are the gametes here? Capital V, small v. Here we have small v. Let's do the cross. Let's do the cross, students. Capital V, small v, small v. Capital V, small v. Capital V, small. Capital V, small v. Right? This will be small v, small v. This will be small v, small v. Small v, small v. This is going to be your violet. This is going to be white. It's going to be white. So out of the 40 progenies, 50% will be violet, 50% will be white. That is 40 is 50% is what? 20. 20 progenies will be violet, 20 progenies will be white. 2 is the answer. 2 is the answer. Understood? Yes or no? Understood? Yes or no? Or did you go wrong here? See, heterozygous pea plant with violet flowers, it is heterozygous, capital V, small v, was crossed with a homozygous pea plant. Homozygous pea plant. Right? It's violet. So it's white. White, that's why it's small v, small v. Because V is recessive in nature. Recessive in nature. Understood? Recessive in nature. But the white, small v is recessive, that is white is recessive. So we end up getting 50 plus 50%. Understood, all of you? Those of you who went wrong, do you know where you went wrong now? This is the part of where you learn. You can go wrong right now. Examination, you cannot go wrong. Okay? Understood, all of you? This is what learning means. Next question. Students, is the question visible or should I increase it? Is the question visible or should I increase the font? I think I'll increase it. 1 second. That's all I can increase, friends. That's all I can increase. Okay. What is the question? The question is very simple. Question says, in a certain plant, okay, in a certain plants, red fruit R is dominant over the yellow fruit. Capital R is dominant. Capital R is dominant. Okay. Now it is visible, tell me. Now it is visible or it is not visible? Visible or not? Tell me. Visible or not? Quickly. It's blur, huh? Should I write it? I'll write it. In a certain plant, red fruit, that is capital R is dominant. It is dominant over the yellow fruit. Over the yellow fruit. And tallness, capital T, is dominant over shortness. Is dominant over shortness. Dominant over shortness. Now what are they doing? If a plant which is RRTT, capital T, small t, genotype is crossed with RRTT. Okay, your capital T, small t is crossed with small t. What is the percentage of tall plants with red fruits, percentage of tall plants with red fruits, tall plants with red fruits, tall plant with red fruits in the progeny, in the progeny. Okay, all of you visible? Now tell me, visible, right? Now let's do the cross. Let's do the cross. Simple cross. What is the cross, students? The cross is between capital R, capital T, small t, right? A cross is between capital R yes, this is the cross. This was the cross, right? Capital R, capital T, and smaller t. Okay. What are the gametes? What are the gametes you get here? The gametes which you get here is RT and R and small t. Yes, these two gametes which we get here. Here we end up getting only one type of gamete, R and T. Okay, R and T. Now, let's do the cross. RT, R and small t, small r, small t. What do we get? Capital R, capital T, small t. Capital R, small t, small t. Okay. Now this plant here, what will be the phenotype? Red and tall. Red and tall. This will be red and short. This will be red and short. Red and short. Okay, red and short. So, what are they asking you? What is the percentage of tall plants with red fruit? Tall plants with red fruit. What is the percentage? Tell me now. What will be the percentage? Tell me what will be the percentage? Tall plant with red fruit. If this is 100, 50% is tall plant with red fruit, 50% are tall plant with short fruit. Easy. Understood? How do you approach such questions? Do you understand how do you approach the questions? Every single question, a numerical in genetics, always write what is given. See, how do we approach? We approach what is given first, right? Simple cross. Do the cross, form the gametes, do the cross, and just see what you're obtaining. Let's see what you're obtaining. 50%. 50%. That's all. Simple question. Okay. Tell me, friends, if this is visible. Is this visible or should I write again? Is it visible or should I write again? Is it visible or should I write? Visible? Yes or no, quickly? Visible or not? Mary's father, okay? Mary's father has hemophilia, okay? Mary is there. Mary's father has hemophilia, okay? Mary's father is going to be hemophilic in nature, right? Mary first here. Mary's father on top is hemophilic. If he is hemophilic, let's write X, H and Y. X, H and Y, hemophilic in nature. But her husband is also hemophilic. Husband is also hemophilic. What would be the chances other daughter having this disease? What is the chances they're asking you? The daughter having the disease. Beautiful question. Okay, beautiful question. Now, if father has hemophilia, the X chromosome will come. So, can I tell Mary is a carrier now? So her chromosome will be XX and H. Father is hemophilic. Father is the husband is hemophilic. Father is hemophilic. This is the mother. Let's do the cross now. XH Y X and XH. XH X X Y XH and XH and XH and Y. Now they're asking you, Mary's father has hemophilia but husband is also hemophilic. What might be the chance that other daughter having this disease? Tell me, out of the two daughters, what is the chance that the daughter will be hemophilic now? The daughter will be hemophilic. What is the chance now? Tell me. 50%. 50%. Yes. Oh, this is 24%. This is 24%, right? What would be the chance other daughter having this disease? Other daughter, 50%, not 25%, 50%. It should be 50%. Daughter having the disease, right? They're asking between daughter. It should be 50%. What would be the chance other daughter having this disease? Only in terms of daughter, it should be 50%. 50%. Okay. 50% chance that it will be hemophilic in nature. 50%. Next question. Next question. Is this question visible? Is this question visible to all of you? I think it should be visible very easily. How can a female be colorblind? How can a female be colorblind? Go. How can a female be colorblind? Marks in the chat, all of you. Okay. Students, if a female has to be colorblind, understand. If a female has to be colorblind, the mother has to be at least a carrier. See, if for a female to be colorblind, the father should be colorblind first of all, and the mother should at least be a carrier. If the mother is at least a carrier, right? If the mother is at least a carrier, when we do the cross, what do we do? XXXC and XYC. So this female will be carrier. This female will be colorblind, normal male, colorblind male. At least, see. So father should be colorblind, mother should be at least a carrier. If she is a carrier, at least a carrier, then the female can be colorblind. Next question. Which of the following is example of sex-linked disease? Which of the following is an example of sex-linked disease? Go. Following an example of sex-linked disease. Sex-linked disease. What is the answer? Following a sex-linked, not STD. They're not talking about sexually transmitted disease. Sexually transmitted diseases are something different. They're not asking about sexually transmitted disease. They're talking about sex-linked disease. That is only one is going to be your color blindness. Color blindness, students. Color blindness. Apart from color blindness, AIDS, syphilis, and gonorrhea are a type of STDs, sexually transmitted diseases. Understood? Next question. In humans, height is not binary thing. That is, it is either tall or short. It can be a lot of variations where a lot of intermediate are present. This is an example of. This is a example of where we get multiple intermediates in the height. So we have 5 feet 1, 5 feet 2, 5 feet 3, 4, 5, 6, 5, 6, 5, 7, 5, 8, 5, 9, 5, 10, 5, 11, 6 feet, right? So that is an example of polygenetic inheritance. Polygenetic inheritance where we see multiple middle stages. Okay. Next question. Select the correct statement from the one given below with respect to dihybrid cross. With respect to dihybrid cross. Okay. Tightly linked genes on the same chromosome show very high recombination. Students, genes are very close. Recombination will be very low. Genes are very, see, genes are very close to each other, so recombination will be very high. Okay, same chromosome show high recombination. Okay, genes are very close, linkage is very low, recombination will be linkage is low. Linkage is high, recombination will be low. Linkage is high, recombination should be low. This is not a statement. Okay, so linkage is very high, recombination should be low. Okay, genes far apart on the same chromosome, show few recombination? No. If the genes are very far, linkage is very low, recombination should be high? No. Okay, genes loosely linked on the same chromosome show similar recombination as tightly linked ones. Genes loosely linked. Genes are very loosely linked. Okay. Genes are very loosely linked. Do they show similar recombination as tightly linked? No. Okay. Tightly linked genes. Genes are very tightly linked. Okay. That is, linkage is very high. Combination will be very few. Yes. Linkage is very high. Recombination very low. So fourth option is right. Understood? Fourth option is right. Next question. Okay. When a violet flower of unknown genotype is crossed with the white flower, the progenies are violet and white. Progenies are obtained. Okay. In an equal proportion. Then read the following statements and tell which of the following are incorrect statement. Your time starts now. Go. Following are incorrect statements. Which of the following are incorrect in nature? Students, quickly smash the like button first of all. I'll give you 20 seconds, read it properly and then answer. Which of the following are incorrect in nature? Incorrect. Okay. Students, this is called as a test cross, correct? This is called as a test cross. Basically, this is a test cross. Okay. The unknown flower is homozygous. Tell me, is the unknown flower homozygous? If the unknown flower was homozygous, we end up getting all violet. But the question says that violet and white both are obtained. If both are obtained, can I tell the unknown genotype is going to be heterozygous in nature? It's going to be heterozygous because violet and flowers both are obtained. So unknown genotype is going to be heterozygous. So this is a wrong statement. Okay, this is correct. The test is used to determine the phenotype. No, it is used to determine the genotype of F1. Wrong. Okay, wrong. For a test cross to be carried out, either violet or white flowers should be crossed with the recessive parents. No, only violet should be crossed. Why? Because in the test cross, students, we always take what? Dominant phenotype. You cannot take white here. Right? Understood? So this statement is also wrong. You cannot take violet or white. Okay? Should always take violet in the to find the genotype. Understood? So option number 4. Next question. Identify the wrong statement with reference to gene I that control AB of blood grouping. Time starts now. Go. Time starts now. With respect to AB of blood grouping. See, all of you know in AB of blood grouping, we have IA, IB and I. The major difference between all three is that the sugar. Major difference is going to be the sugar, right? IA and IB and IA and IB both have different types of sugar. Small I doesn't have any sugar. Okay. IA and IB are present together. They express same type of sugar? No. They express different type of sugar. This is the right answer. That is the wrong statement. Option number 1. Next question. A common test to find the genotype of the hybrid is by to find the genotype of a hybrid. Crossing one F1 progeny with recessive parent, crossing one F2 progeny with the male parent, crossing one F2 progeny with the female parent, studying the sexual behavior of the F1 progenies. Very easy question. Very easy question again. Very easy question. Test cross, test cross, we just now did. F1 parents with double recessive parents. Just now we did the test cross. F1 progeny with recessive parents. Next question. Given diagram shows a pair of homologous chromosomes during meiosis. Maximum crossing over can occur between. Students, remember, maximum crossing over will happen between the sister chromatids. So what is crossing over? Crossing over is nothing but exchange of genetic material with non-sister chromatids. Exchange of genetic material between non-sister chromatids of a homologous chromosome. Now, when will they show maximum crossing over? If you want to see maximum crossing over, the two genes should be very far from each other. Yes. So can I tell between A and D and this A and D crossing over, maximum crossing over will happen there? Yes. Between A and D and small a and capital D. Option number 4. Option number 4. Maximum crossing over. Yes. Next question. ABO blood group in human beings are controlled by gene I. The gene I has 3 alleles, IA, IB and small I. Since there are 3 different alleles, 6 different genotypes, how many possible phenotypes can be there? How many possible phenotypes? Very simple answer again. Very simple. A blood group, B blood group, ABO blood group and O blood group. 4 different phenotypes. That's all. 4 different phenotypes. Option number 4. Okay, question number 4. Next question. A child has a blood group O. The child has a blood group O. Okay, child has blood group O. The father has blood group A and the mother has blood group B. Work out the genotype of the parents. What could be the genotype of the parents? The genotype of the parents here. Friends, if the child has O blood group, the parent should have one recessive allele. Yes, they should have one dominant and one recessive allele. Very simple logic. If the child has O positive, O group, it should have IA, II. So parent should be, one parent should be, right? If the father has A blood grouping, he should have IA and I. Mother should have IB and I. Yes, very simple. Very simple question. Very simple answer. Now students, this is a good question. This is a very good question again. A very, very good question. Students, do you realize how many questions you are doing today? And these are not direct questions. These are not direct questions. These questions require you to think a little. Okay. See this question now. Andalusian fowls have 2 pure forms, black and white. There are 2 pure forms. Okay. 2 pure forms are there, black and white. If black form capital B WB and white form capital B WB are crossed, the F1 individual appear to be blue color. That is basically a concept of incomplete dominance. Incomplete dominance. Okay. Due to the incomplete dominance, which of the following would be an outcome of the cross between black form and blue form? Answer in the chat. Go. Take 20 seconds and answer easily. Students, if you're watching it recorded, I want you to pause the video and answer. If you're watching it recorded, pause the video, answer the question and then play and skip 20 seconds. That's all. 20 seconds starts now. Tell me once you're done. Okay, take 30 seconds this time. You need this one. This question requires you to think a little. So take 30 questions or take 1 minute today. This question takes 1 minute. Take 1 minute. It's very simple again. It's only a matter of how you do the cross. How you do the cross again. Done? Done, students? Done? Let me do it. Let me do it. Now tell me, what is the genotype of cross between black form and blue form? Black form is going to be capital B, and blue form is going to be capital B and capital W. Okay. Let's do the cross. Here we get 2 gametes, all of you know that, right? Here we get only capital B. this will be completely black is to, this will be completely blue. 1:1 ratio. 1:1 ratio. 1:1 ratio. So answer is obviously option number 4. How was this question? This question, what will happen? Let me tell you. In the urgency, there's a very high chance that you might go for the first option, thinking that, you know, sir, it is a incomplete dominance, so you might go for 1:2:1, but it is not. It is not. Okay. So option number 4. Option number 4. Read the question again. Read the question. In a cross between yellow round and wrinkled green pea plant, yellow round and wrinkled green plant, a population of 800 individuals were found in F2. What would be the number of yellow wrinkled seeds in this generation? Yellow wrinkled. Yellow wrinkled. Yellow wrinkled. Time starts now. Go. Time starts now. Yellow wrinkled. Take 20 seconds, 30 seconds and then answer. Dear students, I want all of you to remember that we did a previous question. Remember, we did a previous question like similar to question we did. 9:3:3:1. Right? Wrinkled, yellow wrinkled. Right? Yellow and wrinkled will come here. Will come somewhere here. Round yellow and wrinkled green. Round yellow, wrinkled green will come here. So what is the answer? 3 divided by, initially how did we do in the previous question? In the previous question, this population was not given. So in the previous question, what did we do? We did 3 divided by 16, that is the total progenies, into 100 we did. Okay. But right now progenies is given. So what do we do? 3 divided by 16 into 800. That is the progenies. What is the answer? Tell me. What is the answer? Oh, you're given already answer. Obviously, option number 4. Option number 4. Next question. A type of gametes formed by the genotype of. Which type of gametes do you observe? Which type of gametes do you observe between capital R, small r, capital Y, small y? Gametes. Very simple question. Tell the answer in the chat quickly. And your marks also. Capital R, small r, capital Y, small y. So what do we do? This to this, this to this, this to this, and this to this. Capital R, capital Y. Capital R, small y, small r, capital Y, small r, small y. Option number 1. Option number 1. Easy question. Next question, friend. Next question. Now, I think we have solved this type of question once already. In a plant, red fruit R is dominant over the yellow fruit. The tallness T is dominant over shortness small t. A plant with capital R, capital, we did this right before. Capital R is capital T, small t is crossed with small r, small t, small t. What is the progenies? The progeny is going to be what? We did this question before. We did this question before, all of you. We will do this question. All of you do this question quickly. Small t, capital R, small t, small t. This was, what is the answer now? All red, yes, all red fruit with tallness, all red fruit with tall is going to be 50% is going to be tall. 50% is going to be tall here. 50% is going to be tall. Done. 50% is going to be tall. Understood? This is going to be tall. This is going to be short. Short. Small t, small t. Next question. Study the pedigree chart given below to identify the disorder. Students, the disease is present, is not skipping generation. Autosomal. Unaffected parents, a parent can have unaffected children. Unaffected parents can have affected children. Autosomal dominant. For autosomal dominant, there is only one example given you in NCERT. Only one example, that is myotonic dystrophy. Myotonic dystrophy. Only one example is given. Next question. Students, next question on color blindness. Next question on color blind. Next question on color blind. Okay. See this question now. In humans, color blindness is sex-linked disease trait located on the X chromosome. A colorblind man marries a woman who is carrier for the color blindness. She's carrier. What is the probability of their children having color blindness? Probability that children will have color blindness. Simple cross. Do the cross. Colorblind, carrier, and normal. So the children, probability of their children having color blindness is going to be 50%. That is, one girl and one boy will be colorblind in nature. One girl and one boy will be colorblind in nature, and the one girl will be carrier, one boy will be normal. So 50% is the answer. 50% is the answer. Now students, such questions are very easy to solve. Very easy to solve. Now students, I want you to solve more questions. Now you'll be like, sir, where do I find these questions? I will give you these questions. I want you to download this particular PDF on the Telegram channel. On the Telegram channel, I want you to download this. On this Telegram channel, I will upload this today only. I will upload this particular PDF today only on the Telegram channel, Basavaraj Sir Biology. I want you to practice more questions. Questions are here. Questions 1, 2, 3, 4, 5, 6, 7, 8. 8 different questions on color blindness. 8 different questions on color blindness I'm giving you. 8 different questions on color blindness. 8 different questions. Now it's up to you if you want to practice or not. It is up to you if you want to practice or just chill in your life. Okay? It will be available to you. Don't worry. Now, what is the probability of homozygous plant? Probability of homozygous plant for both dominant characters. Both dominant characters in a dihybrid cross. That is, in a dihybrid cross. Yellow. Basically, they're asking you, students, basically they're asking you this part. Round and yellow. Round and yellow. It is going to be only 1 in 16. It's going to be only 1 in 16. Only 1 in 16. That's all. Very simple answer. That is going to be 1 in 16. Okay, both are dominant. Both are dominant. Dominant in nature. Okay, both are going to be dominant in nature. Now students, write down few questions. I'll teach you. Write down few questions. Did we miss any question, students? Did we miss any question? I thought I wrote some questions for you, but now, okay. Sometimes they can ask you a question on bar body. Bar body. What is this bar body? Bar body is basically inactive X chromosome. It is a inactive X chromosome. Inactive X chromosome in the female. In the female. So how do you find bar body, students? Bar body can be found with basically X How many X number of chromosomes are there? -1. For example, in the case of a normal female, it's going to be XX -1, right? XX. That is going to be, that's basically how many X's are there? 2 minus 1, that is 1. Okay, 2 minus 1 is going to be 1. In case of Turner syndrome, it is X naught. So X naught minus 1, that is basically 1 minus 1, that is going to be 0. That is called as bar body. That is called as bar body. Okay, that is called as bar body. Now students, there is a concept called as recombinant frequency. Recombinant frequency. Formula is very simple. Number of recombinants divided by total progeny into 100. That's all. Okay, they will give you sometimes in the examination, they'll give you number of recombinants. For example, in this 9:3:3:1, 9:3:1, these two are recombinants. These two are parental. So in this example, if you take, that is basically how many recombinants? 6 recombinants by total progenies is going to be a 16 into 100. That is going to be recombinant frequency. That is going to be your recombinant frequency. That's how you can find it. Okay, that's how you can find it. Okay. Now students, they can also give you a question. Based on number of gametes. They'll ask you find the number of gametes. How do you find number of gametes? Number of gametes is basically 2 to the power n. N is the number of heterozygotes. For example, quick example is very simple. Capital A, capital B, okay. Capital A, capital B, small b, capital C, small c. How many heterozygotes are there? 1 heterozygote, 2 heterozygote. So 2 to the power 2. 2 to the is 4. 4 gametes will be formed here. Okay, 4 gametes will be formed here. Okay. Now students, all they can give you number of offspring. They can ask you to find number of offspring. How do you find number of offspring? That is number of gametes in parent 1 into number of gametes in parent 2. So imagine this is parent 1, this is P1. P2 I'll give you, for example, this is P2. This is P2. Find the number of offspring, tell me. Find the number of offspring now. Find the number of offsprings. This is P2. Quickly tell me the number of offsprings. For example, here, how many heterozygotes are there? Only 1 heterozygote is here. That is 2 to the power of 1. That is basically 2. Okay. Now what do you do? 4 into 2. 8. 8 offsprings. 8 offsprings are there. Understood? 8 offsprings will be there. That's how you find the number of offsprings. Okay, that's how you find the number of offspring. That's all. And that is, students, that's it for today's class. So students, we solved a variety of questions. And I've given you homework question also. I've given you a lot of homework questions to solve. I've given you 3 to 4. Now you might realize, sir, what about the other questions? Actually, in genetics, there are still a few questions which can come. There are few multiple variations of questions which you have done already, which can come. So what we'll do is, when your exam is coming near, I will do a session on all the numericals in biology. One separate session on all the numericals in biology. It could be from your cell chapter, sexual reproduction, flowering plants, again genetics, from your evolution also in your numericals. right? Evolution, ecology, numericals. From all these 5 chapters, we will do numericals as the exam is coming nearby. So by the exam is nearby, you will be able to solve more questions. Okay. So students, that's it for today's video, and hopefully this was very useful to you. Right? You understood where you can go, where you can go wrong, and where you can tell it correctly also. And I've given you homework, all those questions. So in today's video, in the comment section, I want to see all the marks. What marks did you obtain? All the students who did recorded also, tell me your marks in the comment section. Okay. So with that being said, thank you so much students, and I will see you in the next class. Okay. Bye bye, take care. My next class is going to be on Tuesday. Okay, next class is going to be on Tuesday. That is cell cycle, cell chapter part 2. Okay. So take care students, bye bye, and have a great day ahead. And students, in the description, if you go, there will be certain link which is very special link. Go find it out what it is. There's a special link in the description. Go find the special link, the first one. Surprise.